Hi everyone, my name is Michael. I'm from Shenzhen, China. I'm currently a senior at the Stony Brook School in Long Island, New York, and today I'm going to present about my research paper, The Potential Applications of US mRNA Vaccines in China and Implications for a Universal COVID Vaccine. Starting with the introduction, the COVID outbreak starts in 2019, Wuhan, China, and then in 2020, there are two major dominant strains that was found, which are alpha and beta strain in UK and South Africa. Then in 2021, there is the Delta outbreak. In 2022, there is the Omicron outbreak. And then 2023, currently in China and some of the Asian countries, there is the XBB recombinant outbreak that brought a new wave of COVID cases. And here is a graphical representation of the biweekly cases per million of China, US, and Japan. And you can see that there are some huge spikes starting in December of 2021 all the way until the end of 2022. And that specific period is the Omicron outbreak, and that is my focus of my paper. Moving on to the vaccination types, here are the list of the currently used vaccinations for COVID-19 from US and China, and we can see that in the US, there's a variety of vaccines, such as mRNA, the protein vector, and viral vector, and they all showcase a very high efficacy rate. Specifically, the Pfizer and Moderna, they have an efficacy rate in the high 80s and even in the 90s. But however, looking at China, China is using the inactivated virus vaccine. And the most common vaccine in China is the Sinopharm. But the problem is that it has a very low efficacy comparing to the mRNA vaccines developed in the US. And that is like the focus of the paper. So the problem is that in China, people developed the Sinopharm vaccine during the Delta outbreak, and they used that vaccine for the Omicron and the XBB recombinant outbreak. And then before the XBB outbreak, you can see that the China have very low number in the COVID cases due to the very strict government zero COVID policy. But in 2023, once the policy was removed, people found that they are unable to contain infection and COVID cases, which means that the Sinopharm is ineffective against the newly mutated strains. Even though that most of the Chinese populations are vaccinated with two dosage, they are still getting the COVID once the policy is removed. So that's the big problem. And then looking at the US, they have various types of COVID vaccines with a very high efficacy rate, such as Pfizer and Moderna with the mRNA vaccines. Also, they are still improving on their vaccination, such as the newly developed bivalent booster in 2023, also known as Omicron targeted vaccines. So this booster shot specifically targets the BA4 and BA5 Omicron variants. So in this paper, my research question is I want to see if the US mRNA vaccine is applicable in the Chinese population in order to combat the current outbreak. In this paper, I used the NCBI database of SARS-CoV-2 virus in order to retrieve all the nucleotide and protein sequence from of the COVID-19 of the very specific strains. And then I used the blast comparison from the NCBI to compare the nucleotide and the protein sequence and to identify specific point mutations that might cause the change in the virus and their infectious rate. So in the comparison, I specifically targets the spike protein on the virus. So the spike protein on the COVID-19 is also known as the surface glycoprotein, and it is a transmembrane fusion protein that plays a crucial role in binding the receptors of the host cells and initiating the infection. And the table on the right shows the specific like regions of the spike protein, and you can see that there are the S1, S2 region. So the S1 subunit is responsible for recognizing and binding to the receptors of the host cell, and the S2 subunit is responsible for fusing the envelope of the virus with the host cell membrane. In the blast comparison, I first target the BA 2.1, 2.1 Omicron variants, and then I chose 6 different regions as the subjects of my comparison. So first, China, obviously, it is my subject of this paper. And then I chose Japan as a comparison subject because they're very similar to China in the virus strains, and but they use a different type of vaccination than the Chinese Sinopharm one. And then for the US, I chose 4 states: California, New York, Washington, and Arizona as my subjects of comparison because I identified the most mutations and differences in their nucleotide sequence when comparing to China. So I believe that they have the greatest potentials for showing regional mutations and variation, which will complicate the problem. And then in the comparison process, I cross blast the spike protein sequence of each region. And for each of the region, I chose 10 samples as like representation of the entire region. But however, for China and Japan, I was unable to obtain 10 different samples. And for China, I only got 2 strains. And for Japan, I only got 4. So that's all data from the Omicron outbreak. And then in the comparison process, I recorded the percentage of alignment and specific point mutations in the cross comparison. And then I calculated the average and standard deviation percentage for the comparing regions. So here are the results from the spike protein comparisons. This is a heat map of my blast results, and we can see that there's very high percentage of alignment, that each slot has a value above zero point 0.9, which means 90%, more than 90% of the spike protein sequence are aligned with each other. And a value of 0.99 also means 99%, means that only a few amino acid difference between the 2 strains in the comparison process. So we may conclude that the China, Japan, and US strains are very similar in their spike protein, which could be an indication for the high likelihood of the application of the US vaccines, since all the strains are very alike to one another. That means that the the mRNA vaccines that would be effective in the US population would also be effective in the Chinese population. And then looking at the point mutations, this is the data table that I recorded each specific point mutation that appeared in each individual blast comparison. And then that the mutations recorded were like very common and appeared for more than 90% of the blast comparison. And we can see that the most common ones were the N161K and the G679R point mutation in between China, Japan, and the US states. And when looking into the positions of the point mutation, I found out that they are not all in the functional region of the spike protein, but in their structural purpose, which means that they are not that significant on affecting the virus into a different strain. But however, that is very good evidence for fast mutability of the virus, and that may affect the vaccine efficacy of the US and the Chinese vaccines. So we need more studies on that in order to obtain a firm conclusion. In my second part of the blast comparison, I blast against the latest recorded sample in China with the general BA4 and BA5 samples using the same method as the BA 2.1, 2.1 samples. Why I did that? Because the newest developed vaccine in the US is the bivalent booster, and they only targets the BA4 and BA5 variants. I I want to see that would it work against the current outbreak in China, and should we develop more vaccines for the rapidly mutating virus? And then for subjects of comparison, I retrieve all the samples for both BA4 and BA5 from the US, and each are from a different state in order to consider the high variability and coverage for the entire country. And the latest samples in China were all recorded in 2023, indicating the earliest, the latest outbreak. Here are the results from the spike protein comparison between the latest Chinese sample with the BA4 and BA5 samples. And for the Chinese sample, I used two different ones. The sample starting with WKL is a strain from the FY 3.1 variant, and it is recorded in May 6th of 2023. And the second one is the WHA strain, which is from the BA 7.14 strain recorded on January the 2nd of 2023. And then from the comparison from the table, we can see that all on average, all the alignment exceeded 0.95 or 9% of similarity with their spike proteins, which is higher than some of the alignment between the regional BA 2.1, 2.1 strains. However, even with that high percentage of alignment, we cannot guarantee the certainty of the effectiveness of the bivalent vaccines on the current outbreak in China, but this uniformity will be a great indication for the likelihood of the application of the vaccines. And then, in order to make a firm conclusion, we would require further studies in order to consolidate the results. In conclusion, even with the reduction of lethality but increase in transmission rate and mutability, SARS-CoV-2 virus is still a major public health concern. In this paper, I discovered the low regional variability of dominant strains in China and the US, and which gave me a better understanding about the applicability of the US mRNA vaccines in the Chinese population. Also, I foresee the potential of overlapping between the influenza and the COVID outbreak in the future. And currently, scientists are developing a so-called universal influenza vaccine to target all strains of the flu. And I'm thinking that that idea, that concept, we can use also use it on the COVID outbreak and develop a universal COVID vaccine due to the fast transmission rate of the virus, and also that not all countries have the ability to manufacture their own vaccines in time to combat the virus. Lastly, throughout our study, I found a lack of data in the Chinese population and Chinese strains. So, in order to improve on the study, We need more sequencing and public health data in order to make a firm conclusion, and maybe need some clinical trial experience and research in order to make a final conclusion regarding the applicability of the US mRNA vaccines on the Chinese population. That's the end of my presentation. Thank you very much.